Friends, if the individual components of a mixture have widely dissimilar physical and chemical properties, it is very easy to separate one from another. But as the components get more and more similar, then it becomes increasingly difficult to separate them. For example, most of the amino acids are closely similar in their physicochemical properties. Because of this, it is not possible to separate one amino acid from a mixture of several by a conventional separation method such as fractional crystallization. However, separation can be achieved by chromatography. Hello and welcome to some biology class. Friends, today we shall discuss about chromatography. Chromatography refers to a family of closely related powerful separation methods. The first detailed description of chromatography was given by Michael Sweat, a Russian biochemist who in 1906 separated chlorophyll from plant pigments. The term chromatography is derived from two Greek words. Chromo means color, graphy means to write. All chromatographic systems consist of two immiscible phases. They are stationary phase and mobile phase. The stationary phase can be a solid or a liquid or a gel, or it can even be a solid liquid mixture. Whereas the mobile phase can be a liquid or it can be a gas. The choice of the mobile and stationary phase is made in such a way that the components of the mixture to be resolved have widely differing partition coefficients. The mobile phase either moves over the surface or percolates through the interstices of the stationary phase. The sample introduced into the mobile phase undergo repeated partition between the two phases while being carried out through the system by the mobile phase. Different components of a sample mixture interact with two phases differentially on the basis of small differences in physicochemical properties. This differential interaction results in differential migration of individual components. The component which interact strongly with the mobile phase and interact least with the stationary phase migrate faster, whereas the component that shows least interaction with the mobile phase and interact strongly with the stationary phase move slowly. This differential migration of different components through the system is responsible for their ultimate separation from each other. The basic principle of all types of chromatographic methods is distribution coefficient. It is also known as partition coefficient. It is used to describe the way in which a solute partitions itself between two immiscible phases. After entering into a chromatographic system, a solute immediately distributes itself between the mobile and the stationary phase. If at a given time during chromatography the flow of the mobile phase is stopped, then the solute will be in equilibrium between two phases. The term partition coefficient describes the relative concentration of solutes between the mobile phase and the stationary phase. This coefficient is denoted by the letter K, which is equal to Cs by Cm. Cs is the concentration of the solute in stationary phase, whereas Cm is the concentration in mobile phase. For example, if the partition coefficient is zero point 0.2 for a substance between a cellulose stationary phase and carbon tetrachloride mobile phase, then the concentration of the solute in carbon tetrachloride is 5 times than that present in the stationary phase. On the basis of principles, chromatography can be divided into 5 different groups such as partition chromatography, adsorption chromatography, ion exchange chromatography, gel filtration chromatography, and affinity chromatography. First, we shall discuss about partition chromatography. In this technique, separation is achieved by huge number of partition steps that take place on microscopic granules of a hydrated, insoluble and inert substance such as starch or silica gel, which is packed in a column or layered on a plate, or the granules of cellulose in the paper. The granules, though inert but are hydrophilic. Because of this, they are surrounded by a tightly bound layer of water molecule which is immobilized. This immobilized layer of water acts as the stationary phase on which flow the mobile phase of an immiscible solvent containing the mixture to be separated. As the mobile phase flow, the sample components undergo huge number of partition steps between the stationary phase and the mobile phase, as a result of which they are separated. The second one is adsorption chromatography. Adsorption chromatography is based on the fact that substances differ in their adsorption-desorption behavior between a solid stationary phase known as adsorbent and a mobile phase which is a liquid or a gas. Adsorption is a surface phenomenon. Because of this, the concentration of a substance is greater at the interface than in the surrounding medium. The components which interact more with the stationary phase, that is the adsorbent, and less with the mobile phase are retarded to a greater extent, as a result of which they move slowly, whereas the components that interact less with the adsorbent and more with the mobile phase migrate faster through the chromatographic system. This differential migration of components on the basis of differences in adsorption-desorption behavior is responsible for their ultimate separation. The third type is ion exchange chromatography, which is defined as the reversible exchange of ions present in the solution with those that are electrostatically bound to an insoluble support medium. Ion exchange chromatography is of two different types: cation exchange chromatography and anion exchange chromatography. The ion exchanger consists of an inert support medium with which either positively charged functional group in case of anion exchanger or negatively charged functional group in case of cation exchanger are covalently bound. These covalently bound functional groups are electrostatically bound with oppositely charged ions. For example, in case of anion exchange chromatography, negatively charged sample components that are present in the solution will interact more with the stationary phase. As a result, they will be retarded. In this case, there will be an exchange of negatively charged sample components that are present in the solution with the negatively charged ions that are electrostatically bound with the anion exchanger. As a result, they will be retarded. But the uncharged components and the positively charged ions will be not retarded to the same degree. As a result, they will be eluted out fast. So ion exchange chromatography is based on the principle of reversible exchange of ions. The next one is gel filtration chromatography, which is based on molecular size. In this technique, the support medium is a gel which consists of porous bits and the pore size is strictly controlled. So the molecules which are smaller get entrapped in the pore. As a result, they are retarded, so they move slowly. But the macromolecules which are larger than the pore move unrestricted through the system. As a result, they are eluted out fast. The last one is affinity chromatography, which is based on the specificity that exists between an enzyme and its substrate, between an antibody and its antigen, similarly between a receptor and agonist, and also between an enzyme and a substrate analog. For example, when the gel matrix is coupled with the substrate analog and when the cellular suspension containing the enzyme is allowed to percolate through, then the enzyme will bound with the substrate analog. As a result, they will be retarded and they will be mobile, whereas the other components which do not interact with the substrate analog will move faster and they will be eluted out. So friends, this is about chromatography and its principle. For more updates, please subscribe my YouTube channel, Sam Biology Class, and also please don't forget to share your comments. Thanks for watching.